the lecture of yesterday, that was on the probe in general, and then I will start with and I will also do the labeling in protein, so the fluorescence probes for protein. So that's in these 2 sessions I will be able to complete these 2 parts.
Yes, perfect.
Okay.
Yeah.
Good.
Yeah.
So
So
I will
Simran,
share.
introduce. Simran will introduce
Yes,
first.
sir.
Yes, yes,
Okay,
just
so very good afternoon, everyone, and to our Dr. Lisa. I welcome you all again to our web course, and I hope you all are you're finding these lectures really informative and insightful. So I would like to introduce you all again to our speaker, Dr. Aliza Bombada, and I please request ma'am to kindly deliver your talk.
Okay, so thank you for the introduction. And so, as we said also yesterday, so I will complete the lecture on the probes, fluorescence probes for biomolecules, a general view on bioprobe probes, fluorescence probe of biomolecules. Sorry. Okay, so yesterday, so we ended with introducing the molecular rotors, and that are probes that can be very useful for detecting local viscosity in biomolecular sample, also in cell can be done. So, I mean, with this sentence, I would like just to remind the general criteria, let's say, that with which we are choosing for probes. So we have to choose, of course, the probes that is able to detect the properties that we are interested in. And the chemists that develop this probe, they have this also in mind. So then they try to design molecules in order that they have that with their chemical reactivity or with their molecular structure are able to detect, to change their fluorescence in function of change in a specific properties of your system. So, okay, so an example exactly of another type of probe that is in the category of the fluorogenic probe is a donor dark quencher dual probe that we also see yesterday when we were discussing about resonance energy transfer. So as we said yesterday, so that's the probe is composed of a fluorescence donor and a quencher acceptor. So that means that is a molecule that is not fluoresce and whose absorption overlap with the emission of the donor. That's what we are. What is required for transferring the energy by resonance. Okay, so it's not only the overlapping that is important for transferring the energy by resonance, is also the distance that play a role, as we were seeing yesterday. And in fact, that is also how this probe is working. So normally is done is a way of labeling a substrate of an enzyme, in particular protease, that, as we said, that their function is to cleave is in a specific site. So normally the protease, they are peptidase, so they cleave the peptide bond of in between two amino acid. Okay, so when the substrate is intact, the probe is in the closed form, and the quencher is close to the donor enough in order to quench completely the fluorescence of the donor. So no signal. Then the enzyme comes, does its work, and cleave the substrate, so the donor and the acceptor are far apart, and the fluorescence of the donor is restored. So far, so good. Is in the biochemical process, so this enzymatic reaction occurs in the so-called active site of an enzyme, which is like normally is like in a pocket of the enzyme. So the substrate goes there, binds, and the chemical reaction, because is a chemical reaction, occurs, and then the product are released, so they move out from the active site of the enzyme, and the active site then in the enzyme, which is a protein, is free again to welcome a new substrate, and then the reaction, then the product are released, and so then again a new substrate, and so on. So the enzyme works in cycle. That is actually the major characteristic of the enzyme catalysis, are catalysts. So they are just there to make a reaction faster, and so to help the occurring of this reaction. Okay, so this is actually fundamental, because as I said, the product need to be released again in the environment. And so then we come to some problematic that can exist also in the design. So in the design of probe, because you have to take care about this. So enzyme works in aqueous solution. That's our biomolecule, normally in aqueous solution. So that means that the product has to be, let's say, hydrophilic enough. So that means for being released. So that is they have not to be scared to go back in the solution. Actually, they have to be they have to be favored to go back to the solution, because if they don't do this, they don't free the active site, and then the enzyme is blocked. And so this was a problem that we encounter in designing such dual probe that I just described. And so so as you see, so then we wanted to do this. We wanted to design such probe, and we get inspired by the literature, and we saw that the quencher, so that is the quencher part, is Dapsil, which is a very common quencher molecule, and so we also use this as a quencher. And in our case, it didn't really work. So actually, we were seeing after some time that the cleavage of our substrate was not complete. And so after study, so we went to we were testing and also with other method, NMR and so on. So practically the conclusion what we observed is that the product actually was not released. In particularly, this part of the probe was not released after cleavage and was blocking the active site, and the enzyme could not work anymore. And this is also understandable, because if you look, this molecule is quite hydrophobic. And so it doesn't like when to be in the solution, especially when you cleave. So then the entire molecule. When the substrate like this it can get some, let's say, maybe there are some charges also related to the amino acid, which and so can be soluble. But the dapsil alone after cleavage, it's not as soluble, so problematic. So that's what was inspiring us, or motivating us, let's say, to make an analog of dapsil, which is this molecule. And so we design a new dark quencher, which actually we patented it also because it doesn't exist. And so it's this one here that this you see is very similar to dapsil. So it's actually the scaffold is the same, but with the OH, additional OH group is hydrophilic. It becomes hydrophilic. And so we were testing this new dark quencher and compare it to dapsil using this substrate, this particular substrate, which is specific. That is the cleavage site of thermolysin, which is an enzyme that cleave between phenylalanine and serine. And it was already used in the literature with dapsil, and we made the similar experiment with hydrodapsil, and all the tests that we did, also with thin layer chromatography, prove that when an hydrophilic quencher is used, the enzyme actually works more efficiently. So that is this story indicates that the design of probes is not so simple and straightforward. But one thing to say, I want a fluorescence probe because I want to see something. Yes, okay, that's good. But we should not forget that we are working with a biomolecular system. And the biomolecules have their own exigencies, and when we want to study them, we cannot interfere with their exigency. We have to respect this, and we have to try to adapt to their exigencies. And only in this way we can we have the good tools for investigating such system. Okay, so yes, so that is, I also put the publication because, of course, I cannot enter in all the details of this work because, unfortunately, lack of time, we see. But if you are interested, you are so welcome to look in this paper, and maybe also to consider hydrodapsil for your research. Okay, so yes, I was not putting yes in the full screen. So another example of probes, so are the solvatochromic probes that allows ratiometric sensing. So in contrast to fluorogenic reporters, which we saw are characterized by an increase of fluorescence, or let's say off-on. So then switching, practically fluorogenic reporters are switched on by the mechanism that we want to study. Instead, the solvatochromic probes enable a ratiometric sensing, which means that they are probes that change the wavelength of emission as a consequence of the mechanism of the event that they are monitoring. And that may have some advantage in some cases. So here, for example, is the comparison of two calcium indicators. So the fluo4 that we just we are talking about is a fluorogenic probe. So then when calcium binds to the sensing, to the targeting, to the part of the molecule that is sensing the phenomena, is getting fluorescence. So and this, you see, this the intensity of that we can measure is increasing with the concentration of calcium, and just like this. Okay. Instead, in the one is a solvatochromic probe, and so here is practically the same experiment. So the concentration of calcium, and as you see, there is a shift of the emission. The color of the probe is changing in fact when it bounds calcium, and of course, comparing the height of this band, we can determine how much calcium is there after, of course, calibration. And so what is the advantage is that this signal, so the shift between the band shift is actually independent of the probe concentration. So what instead depend also of the probe concentration is the intensity. So that means that when you have an intensiometric probe, you have to distinguish, so you have to be sure about the concentration of your probes that you have in, because you have to compare always what you can compare. So you want to just separate the effect. So you are interested in the increase of fluorescence due to the calcium binding and not due to a change in concentration of your probe. This problem, you don't have it here, because the shift in the wavelength is the same independently of how much probe you have inside. And so, okay, so then in this case, you can also, let's say, overcome problems due to an incomplete level. So that means that you may have the sensitive part that is not leveled with the probe, and that doesn't provide signal. And in the intensiometric probe, that would mean that calcium is not bound to the coordinating part, but instead it's not true. It's simply that you don't see it because it's not leveled. So that says instead here, okay, if it happens that you have the coordinating part unlabeled. Okay, you don't see this, but you can see all the others. And the shift remain the same. Okay. So another, yeah, that's are also some other probes. So that is the solvatochromic probes that change their color, as I said, as a response to the change in the local environment. And the classical solvatochromic probes are based on push-pull fluorophore operating by internal charge transfer mechanism that we saw, that is the mechanism that mainly affect fluorescence. And in this case, actually, obviously, because as you remember, and that is a probe actually that detect polarity. So the change, as you see, it change its color in function of polarity. And so that's, of course, the charge transfer is a fundamental phenomenon, because that means that if the fluorescence is associated to a charge transfer, it's clear that the dipole of emissions of the dipole in the excited state is changed with respect to the one on the ground state. The solvent need to be rearranged around probe, and that is what is and this relaxation is as a consequence a shift of the wavelength towards red color. Okay, so and so that's, yeah, that is an example, and the Nile red is one of is the most famous of such type of probes for detecting polarity. And so that is also as evident is a parameter very much important, because, for example, also inside the cell you would like to detect local polarity regions that are more polar than others, or something in a membrane where the polarity is less with respect to the cytoplasm, and so on. So that's problematic that you may have, and it's nice to know that the probe of such kind exists. Yeah, finally, another parameter. So as we were talking about fluorescence lifetime as the other after intensity is the other fundamental characteristic of the fluorescence of the fluorescence process, which is the fluorescence lifetime is the time, the average time during which the molecules stay in the excited state. And this time can be affected, reduced, or prolonged according to the occurring phenomena of the surrounding phenomena that happen. And so that's why lifetime can be also used by as a parameter to be detected. And so there are probes that are designed to be sensitive on that in this respect. And for example, so here is an example also of a molecular motor, and it's the different the decay, how the decay, the fluorescence decay get slower, and so that means that the fluorescence lifetime get longer in function of viscosity. You see, that's a color. If you follow the color code, that is the viscosity scale in the centipascal, and that is the fluorescence lifetime, of course. So flatter is the decay, longer is the lifetime. Here we are in the logarithmic scale, so that's why it's linear. Or as we say, just to remind some concept, the decay is normally exponential. And in the logarithmic scale, you get linear. Okay, so what I want to say, yes, so another story, another point is it can be, so fret also affect, as we said, fret affect the lifetime, because it introduces a new disexcitation pathway, or let's say a new pathway that deplete the excited state. Okay, so and following the lifetime is the basis actually of a technique that you most likely heard about. It is called FLIM, and that means fluorescence lifetime microscopy. So with microscopy we can work in cell, and so that is just one example benefiting of the fret between two molecules. So here is practically, just briefly in this picture, you can see the interaction between these two proteins, the NMT1 and the UHFR1. And so, in this case, is the aim is to find a medical compound that how to say, interfere with the interaction of these two molecules. That's because these two molecules are involved in the proliferation of tumor, and so then in principle we would like to avoid that they interact. We would like to interfere with this interaction. So let's look for compound that do this, and so we'll see if. And so several compounds were tested, and the one that is called the UM63 could be selected because of FLIM experiment. And so how is working? So here, for example, you have the two molecules are that are labeled with a fluorescence protein. So EGFP1 and mCherry, the other one. So fret occurs with these two fluorescence protein. We excite EGFP, and if a fret occurs, we will see the fluorescence from mCherry. So we excite EGFP, and in principle we should see green, which correspond to the fluorescence of mCherry. And in this case, instead, in A, there is only the donor, so there is only EGFP. There is no the acceptor part. So of course we see the fluorescence only of EGFP. So that is just the comparison that we can have. So let's say no fret here and fret here. The color actually is not the intensity, is not the real color, but actually is a color code with which we can represent the lifetime. So here you have the scale. So you see the 2.7 is nanosecond, is the lifetime expected from his GFP, and is the blue one, and the green will be something in between these two extremes, and is the lifetime expected for mCherry. So that's the color. As I said, I repeat, is not intensity, but it, let's say, color, give a color for visualize a particular lifetime that was detected in this position. Good. So that is our scale, and that we get to follow. So no fret. And now we add this compound, and we see that when we add this UM63, we see only the lifetime, practically only the lifetime of the donor. So that means that there is no interaction between these two proteins, because they are they cannot get close enough for allowing fret, which is instead the case here. So this kind of experiment was useful for deciding that the compound that was added in C is better for such purpose with respect to the other one. And so how to use fluorescence lifetime and labeling with fluorescence protein. Okay, so I see that with this this overview about the general probe can end. Just some conclusion, that let's say just to fix the concept about the fluorogenic probes, where we follow intensity, and that in principle they are easier to design, and they may allow lifetime imaging easier. Solvatochromic probes, they have the other characteristic that they are they can sense, they can enable, actually this S should not be there, sorry, enable ratiometric measurement. They are a little bit more complex to design, and lifetime imaging is possible also, but of course interpretation is maybe a little bit more difficult. But yeah. Okay, so that's a summary, let's say, table for this section. And in the next, I will start to talk about probes that are used when proteins are the target of our investigation. So while I'm changing now from one lecture to the other, in case there are questions, don't hesitate.
So do you have any questions up to now?
Okay, so then let's go on. So yes, okay, so fluorescence probe for protein. Before entering really in the subject, I just make a very short overview about what protein and actually biomacromolecule are. That is something that is important to keep in mind. And so here I represent the molecule that we called biopolymer. And why biopolymer? Why they are, in fact, so bio because they are biomolecule, but polymer because they are done by practically, let's say, copy of building blocks. So they are done by building blocks that we call normally monomer, and you have one building blocks that you have many, so polymer. And so, yes, the building blocks in protein are the amino acid, and the building blocks are the in nucleic acid, DNA and RNA are the bases. Okay, so just an important also, as we will see, because as also, I mean, what I'm saying, I know biochemistry, okay, it's but as also some consequence in the probe, not only because there are mechanisms that we would like to study sometimes, but also there are mechanisms that we will use during the to allow the labeling process, as you will see. So that's why I make this short overview. So the nucleic acid, they have the informational task. They provide, in particular, DNA as a genetic material. Then is the RNA is practically the message that is allowed to transfer the message in order to synthesize protein. So they are actually related in this molecule too. And the protein have the operational function, so are the factory of life. So they do everything in our body. Okay, so as I said, polymer, so polypeptide. Proteins are polypeptide. That is the building block of protein, is the amino acid. And so this part here, the amino group and the carboxyl group, are common in all the amino acid. They just differ by the side chain, which is represented by R. Here an example, this is glutamate, that is lysine, as you see. So the amino and carboxyl group are the same, and just the side chain that change. And to create the chain, so the building block has to be linked, and the linkage is the peptide bond. That is practically an amidation that you have here. Okay, so, and here there are the list of all the natural amino acids that we find in protein. Some are polar, some are non-polar, some they can be charged according to and plus or minus. Now, of course, accord is a consequence of deprotonation. So the charge depends on the pH, if it's deprotonated or not. But okay, that's are the major characteristic, let's say, of the amino acid. And among them, there are three that we already encountered in our discussions that are fluorescents. And tryptophan, phenylalanine, and tyrosine. So nucleic acid, so just nucleic acid, the building block is the nucleotide. And so here the common part is this one, is the sugar and the phosphate, and then what change is the base that are the 4 base. So the amino acid are 20, and the nucleic acid are 4, only 4. In DNA, adenine, guanine, thymine, and cytosine. Instead, in the uracil, thymine is substituted by. So, in RNA, sorry, you have uracil in the place of thymine. Okay. So another difference between DNA and RNA is that the presence of this oxygen here in position 2, which is there in RNA, but not there in DNA, which is deoxyribonucleic acid because of the absence of this oxygen. Okay, so here is the typical double helix that we have in DNA, and so you see the base interacts with the hydrogen bond, but the chain is here, and that's is the chain which, so we practically, this hydrogen bond are important for secondary structure, but to form the polymer, you have this backbone here that form with the glycosylation. Okay, so now we have our major polymer, but of course the structure that you have to keep in mind, because as I said, you will see we will always refer to this characteristic to see what can be done and what cannot be done, or how we have to interpret one signal in our investigations. So, yeah, that's also another point that is a difference that we have to keep in mind. In nucleic acid are structurally, let's say, simpler than protein. Protein, there is really a great variation of structure that we can have. Instead, the nucleic acid, let's say, a little bit more simple. So, of course, the double helix is the structure that is most common. It's not completely true what I'm saying, you will see, so the sense that there are also different structure in nucleic acid, but let's say the variety is less than in protein. And then this hydrogen bond that's the so-called Watson and Crick base pairing, are really extremely important for structural purpose and stability also. And as you see, you have an adenine can face only a thymine, and the guanosine only a cytosine. And that, so the molecules are complementary in that way. And if in case, let's say, for some reason, for a mutation or something, a structural change, or interference, whatever, adenine is in front of a cytosine, but there it's called a mismatch, because this hydrogen bond cannot form properly, or are not formed completely, because adenine can form only 2. Cytosine would like to form 3. It has group enough to form 3, but since it doesn't have the complementary group in the adenine, then it's remain this third hydrogen bond cannot form, and so there is a stability, so can affect the stability. That is practically what a mismatch is. Okay, so as I said, let's go also in this what is similar, and so then this biomolecule, they have a certain similarity also in the way in which they assemble, in the sense that we can recognize the so-called primary structure, which is a chain of amino acid, is just the sequence practically for the protein, and the sequence also for the nucleic acid, a chain of base. And then they organize in a secondary structure the protein, the alpha helix, the beta sheet, and also the amino acid, for example, the double helix, the stem loop, etc., etc. Then you can have a tertiary structure, so then a three-dimensional. So it's how you really, in space, how the structure, the secondary structure is organized. And also the so-called quaternary structure, when you have a complex of different molecules. So you have it in protein, and you can also have it in nucleic acid. And you can also have, actually, which is also relevant in our research, you can also have a hybrid quaternary structure. That means that you have protein and nucleic acid in complex together. Okay, and so to conclude, I was saying that these biopolymers are related, and so is the central dogma of my molecular biology that show how we can synthesize a protein starting from the information provided by the DNA. And so to make a complex story simple, that's the first step is a transcription, where practically the message of included in the DNA written with the alphabet of the DNA will be written with the transcribed practically. It's not, actually this is not really a translation, is more, that's why it's called transcription, and also is a way of how to distinguish between the two fundamental process, because let's say that is just to copy it. It's practically the language is the same between the DNA and the RNA. You have the 4 bases, and so you have just to copy the message. But afterwards, the DNA, so there the message written with the alphabet of the nucleic acid, in the RNA, has to be translated in another language, which is which has also a new alphabet with 20 elements, and to arrive to synthesize the protein. And this occurs in the ribosome. And so in translation, so mRNAs are decoded to produce protein. And so this translation, so the ribosome, what is the function practically? The ribosome is to keep together, right, to facilitate the coding by inducing the binding tRNA. So the RNA are several, and so type. So one that brings the message, then the other is that has this codon. So anticodon, actually, that is complementary to the codon of the presence in the RNA. So practically, the ribosome read the RNA and call the tRNA that has the right complementary sequence. So the codon is just are 3 bases. Is a code made of 3 bases, and that's what the tRNAs, the transport tRNA, what the tRNA actually transport is the amino acid. So it has so it comes actually from here. Sorry, I was. It comes from here, and it brings, so it has linked an amino acid. So then practically, okay, read the sequence, yes, okay, the codon, and it say, okay, it's like to call the appropriate amino acid to form the sequence of the protein. Okay. So, and the amino acids are chained together, and the protein get formed. And then, of course, there is a dissociation of the tRNA. Good. So, it's clear by all what I said. So it's very, very short overview of essential mechanism of life, practically. But enough to get aware about the importance of this molecule, and of course, and also the fact that the interaction between protein and nucleic acid is fundamental. Then also conformational transition, and because they play key role in all this process. And then we said that protein have several structure, but actually nucleic acid too. That's I said, yes, okay, they are more simple. They are a simple alphabet. They make less type of structure, but anyway, many too. So there is a typical alpha helix, but you see, you can have some cruciform, you can have some triplex also, the hairpin, you can have quadruplex, and so yes, and all this structure have a function in biology, and so it's interesting to have the possibility to study them, to see how they form, or maybe to find a way to favor their formation or to hamper their formation according to a particular therapy. And so the basis of this is to know them. And the structural methods like X-ray, NMR, cryo-electron microscopy, of course, are extremely important and provide valuable information, but they are not really ideal to investigate the dynamics. So they give maybe a snapshot of a story, but it's difficult to follow the kinetics. To this respect, fluorescence technique may help, and that's why that's why we are here. That's why we want to know more about fluorescence, and also how we can use fluorescence molecule to study this mechanism, and which fluorescence molecule can be used for one purpose or the other. So, okay, that was, let's say, a common overview for the two major biopolymer of life, also the biopolymer, yes. And so now I focus today on protein, and tomorrow we'll see the amino acid. Okay, so yeah, when we were talking about probes in general yesterday, we were saying that we can distinguish between two categories of probe: the intrinsic in case of biomolecule, you can have intrinsic fluorescence, fluorophore, and you can have extrinsic fluorophore. So let's start to give so I start to give a short overview again about specifically about intrinsic fluorescence of proteins. So we cited along our talks already, but now just remind the important aspect. So yes, protein are already, many of them, let's say, they are already naturally labeled because of these three natural fluorescent amino acid, in particularly the tryptophan, which is whose fluorescence is the most sensitive to the local environment. And it has also a slightly better, brighter brightness. As you see, the quantum yield of tryptophan and tyrosine is similar, but the tryptophan has a higher extinction coefficient. Phenylalanine, it's left a little bit aside because indeed, as you see, the quantum yield is so low and that is not really very much used. But it's important to know that it exists, and actually in some application can be also used, but is not the most common. So another advantage of this fluorescence amino acid is not only the fact that they are fluorescence, but also the fact that they are quite rare in protein. Many proteins has also only one tryptophan. And so that is an advantage because you can say, okay, it's a local probe, so then I can have information around of its environment, and I'm not getting disturbed by many other signals around it. So as I said, the tryptophan has the highest sensitivity to the local environment, and in fact, so we can see is a sort of, we can also use it as in some respect as the solvatochromic probe, as you see, because which is polarity sensitive. That's an example. And in particular, so then you see the color code it helps to read the picture. So when the tryptophan is in the core of the protein, so let's say is more hydrophobic environment without accessibility to the solvent, he has a blue shifted, so this spectra, it's in the blue extreme of his emission band. And that is an example. And then closer to the surface, so let's say more accessible to the solvent, the fluorescence emission shift towards higher wavelength. Solvent is, as you can expect, water, so is polar. So then as soon is polar. So increasing polarity, it's increasing the wavelength of emission. And so this helps us to study phenomena like unfolding, for example, of a protein, or also interaction, because let's say that during an interaction, a tryptophan that is initially exposed may get solved inaccessible because of the interaction, if it's for example belongs to the interacting domain of the protein, and so on. So yeah, okay, that's what we're saying. Classical things is to monitor the protein denaturation, for example, and that's in the unfolded protein. The tryptophan is more easily accessible to the water molecule, and so you have a red shift. Good. So then tryptophan is also very sensitive to some quencher, as we and so we can get some structural information because according how easier is to quench, we may know if the domain in which the tryptophan is exposed or not to the solvent. So we can have some structural information of our protein. And then so okay, what about tyrosine? But tyrosine is, as I said, is less sensitive to the environment than tryptophan. Of course, it's possible to use also tyrosine in many respects, but is less sensitive. So, what often the strategy is, so what often is can be a good, could be a good thing is, for example, can be the tyrosine can be mutated, substituted by a tryptophan, and then you can continue your study. So that is a mutation that should not affect too much the structure, and that's, for example, was done in this molecule that is called calmodulin, that binds calcium, and it doesn't contain tryptophan, but it has several tyrosine. And each tyrosine, one by one, was changed into a tryptophan, and this mutation allowed to study calcium binding. And since, moreover, actually this molecule is a very small molecule, but it's 4 calcium binding sites, and the tryptophan were put in each calcium binding domain. And studying the curve, it was possible to understand the sequence of filling of the different domain. So the sequence of calcium binding to calmodulin, and revealed also interaction between the various binding sites. So just like this. So putting tryptophan in a specific position by changing the sequence of the protein. Of course, has to be done genetically. You have to prepare the appropriate plasmid for doing this, but we are still talking about natural amino acid. So it's in principle should be easier and affecting less the protein. Tyrosine, if we look, maybe we go back. No, wait a minute. We go back to the, yes, here. So this is the emission spectra of tyrosine, and so the actually the wavelength, I'm sorry, is not is not corresponding the wavelength scale, but okay, we can read anyway the spectra. So these are the emission spectra that are the absorption spectra. So then you see, let's say that tyrosine emit between 260 and 350. And in particular, there is a band between 260, let's say, and 300. And this overlap with this part. So you see 260 and 300. It's this part of the absorption spectra of tryptophan. So what could happen according to what we know? That can be resonance energy transfer between tyrosine and tryptophan. And so then we can have a FRET probe, a natural FRET probe in our protein. We can have it, or we can put it by specific mutation. And so, and this what was done in this work, for example, where the tyrosine, so there were some natural tyrosine and some natural tryptophan, but some mutational study with some mutational action, it was substituted tyrosine in one place and added a tryptophan in another one. So to create, let's say, a FRET pair in different position of the protein, and so in order to probe the protein in different position, and to measure, for example, the distance between two specific positions, and so finally to mapping the structure of the protein. So that is a strategy that can be used. Okay, so intrinsic fluorescence, it seems actually quite good, because we have the feeling that we don't change, we change very little or maybe not at all our system. But it has some drawback. Tryptophan absorbs and emits in the UV and is prone to contamination and is photosensitive. Then, for example, the UV is not very amenable to study in the cell. So it's then the brightness of tyrosine is very low, as you see, 200. You saw we were talking about molecules that have several hundreds, thousand of brightness. So 200 is not so much. Tryptophan is just only a little bit better. Then tryptophan has a multi-exponential decay, as we saw, so which requires some efforts in the interpretation. Moreover, has also two overlapping excited states, so has some complexity, let's say, in his own spectroscopy. Then we say that proteins, so they are rare in proteins, but anyway, there are many proteins that have more than one tryptophan, and 2, 3, 6, so it can happen. So as even if it's less frequent than other amino acid, but can a multi-tryptophan protein exist? And that is an additional complication, because we may not know which tryptophan, from which tryptophan the signal comes. And they can have different emissive properties. And so we were seeing some example in the lecture till now, what can be done to distinguish, of course, of all, one option is to mutate the protein in order to let maybe only one tryptophan and then to change the position of this tryptophan along the sequence. That's, of course, could be a possibility. Then other possibility is to make effort in the analysis, like we saw, to distinguish between the lifetime contribution of one tryptophan and the other. For example, we saw this, or by quench study to see, to distinguish between a tryptophan that is more exposed to the solvent than the other. So there are some efforts, but as you see, there are all efforts that sometimes, and you cannot completely, that they are not always completely successful. So drawbacks, so then there are, we can go to some solutions. So then, for example, concerning the story of the brightness and the spectroscopic properties, but some tryptophan analogs has been designed to have larger quantum yield. So you see that is the natural tryptophan, and this is the that is an analog, which is a quantum yield which is actually the double. Then you see the different these are the absorption spectra of the different analogs represented here, and that's are the emission spectra. So then you see there is an analog that may have also an emission in the visible range. Unfortunately, the quantum yield is not very high. So then there is a tryptophan actually, which is let's say in that are the quantum yield in water. Then inside the protein can change, and that's as there is the case of this tryptophan analog, which is actually non-fluorescent, and that's, one would say, okay, what for what can be useful? I mean, I want a signal. It's very similar, actually. Maybe it's the closest if you compare the others to the natural tryptophan. And it can be also useful to have a non-fluorescence probe, or let's say non-fluorescence tryptophan analogs, because of course this is not fluorescence, is maybe not very useful as a probe, but that can be other function. So since it's very, very similar to the natural amino acid, so it can be substituted to, for example, in a multi-tryptophan protein. And you can substitute all tryptophan but one with this analog, and you eliminate the emission for all the other tryptophan, and you retain without changing much in the protein due to the high similarity between the natural and the synthesized tryptophan. So that's how to find benefit also to problematic properties. Okay, so yes, yes, there are non-natural amino acids though. So how we can, so it's not so easy. So it's not we can say we mutate the protein as normally when you just change a natural amino acid. Some efforts to find the proper strategy need to be done. So a way to insert this tryptophan analog is solid phase synthesis, for example, where you practically chemically build up your protein. And this is a technique that's very good, that can be also extended to other analog that are not necessarily amino acid analog, but maybe completely another molecule, because you practically, it's like you synthesize your molecule from scratch. But you can imagine that this method is limited to short peptide. Then concerning tryptophan, it can be possible to make the expression in tryptophan auxotroph bacterial, which is practically bacteria that they have the possibility to synthesize tryptophan, so that they don't have this. And so then the tryptophan analog is chemically linked to the tRNA, associated tRNA, and is added to the sample during protein synthesis, the tRNA, as we saw just before, that the tRNA bringing this fake, let's say, amino acid, but with the code of tryptophan, because anyway, there is not a problem to make a mistake, because these bacteria, they cannot do it by themselves. So then we practically, is a trick in this case. And so in this case we attribute practically the codon of tryptophan to the tryptophan analog, and the synthesis can be maybe done. And another way is to modify the genetic code with the genetic code expansion method, and we will see it later, how it's working, more or less, at least in general. And we will see also that that is also a method to insert also other amino acid analog, not only the tryptophan. So the method number 2 is only possible for tryptophan, but this one is more general. So practically we manipulate the genetic code. And what I want to say, so that are example, for example, of analog of other amino acid that may also be modified in order that they become fluorescence probes. So that is the basis. Here is alanine. And as you may expect for this conjugating system, it's the side chain in this modification get fluorescent, as shown here. Okay, and so yes, we see how can be done with this modification of the genetic code. But practically, so there is an enzyme that is called aminoacyl tRNA synthetase, which is the enzyme that is responsible to link the aminoacyl to a tRNA. That is what is, in order to have to build up, voila, the tRNA here that is bringing, that has this anticodon here to read the codon on the mRNA, and it brings a specific amino acid. Okay, and so is actually is this enzyme will be modified. So here, let's say in lila, in order to link to a tRNA a non-canonical amino acid. This modified tRNA will work in response to a codon UAG on the mRNA, and so it incorporates the amino acid, the unnatural amino acid, into the amino acid sequence. So it's attributed to this transport, so this tRNA, a codon, which of course should not correspond to any other amino acid. If not, the game cannot be played. And so is a code, voila, is a non-occupied code. Let's put it in this way, and so is practically using the machinery of the protein synthesis, but inserting, as a consequence, inserting a unnatural amino acid. And the unnatural amino acid can be designed in order to carry the fluorescence, the fluorophore, or it can contain a group that is compatible with the click chemistry, and then in a second moment the fluorophore is added by click chemistry. So here there are example of amino acid that so let's say, so in this case actually that are not even amino acid with the fluorophore on the side chain. These are practically fluorophore that are the fluorophore is the side chain. So that is practically they are a natural amino acid. So then they have the amino acid, so amino and carboxyl group. So you see it's designed as an amino acid, but is a fluorophore. And all of them can be inserted with this method. These are amino acids that are compatible with the click chemistry, and then of course it's required to add a probe that a fluorophore that can interact with this group, and yes, and you can then label your protein after synthesis. Okay, so yeah, and here we were remaining anyway in the analog of an amino acid, but we can also labeling the protein with organic dyes. We were seeing the hexa, fluorescein, cyanine, etc. And that is chemically done. So organic fluorophore has the advantage that they have a wider spectral range, they have a small dimension, they have a higher brightness than the tryptophan analog that we saw, or other fluorescent unnatural amino acid, but of course they are unnatural, and so then they and maybe they are small dimension, yes, but maybe large, a little bit larger than the fluorescent, the amino acid analog. So that all this can cause the undesired effect that need to be checked. As you remember, the problematic of the hydrophobic quencher was that was quite an issue actually, and so that's all these things need to be checked. Okay. What I want to say is yes. Okay, so I just maybe finish these slides, then we make 5 minutes of break, or 5 minutes, or actually maybe quarter of an hour.
As you convenient, you can.
Yeah. Okay. So yes. So then maybe let's finish here since we have the slides. So then, so how the protein can be labeled with the organic dyes? So, but there are different. So these are the traditional bioconjugation method. And in particular, so the dye can be synthesized with the malamide group. That's something that can be done. It's a chemistry in the lab, so it's okay. Or they are even sold already with connected with the linked with the malamide group, and the malamide group interact specifically with the thiol group, and thiol group exists in protein because it's the side chain of cysteine, and is cysteine, so is in fact a good labeling target in the protein. And the advantage of cysteine is also that they are rare, so then you can label a specific part. And sometimes they can be, in case they are not there, they can be introduced specifically. So in one specific site, and this cysteine is a natural amino acid. Another group that on the organic dyes that can help could be the acetamide, the halogenated acetamide. X is a halogen, in general iode. And this group may interact with also with the tyrosine. Sorry, Ali. So I need maybe really a break with the thiol of the cysteine, and then there are other groups that can be associated to the probe, which interact with amine. That will be and amine, where amine can be found in protein, but amine can be the side chain of lysine. A drawback is that lysine is more frequent than the cysteine, so then yeah, we have to be careful because of this. But amine can be also in the N-terminal group of a protein, and which is unique, and that could be an advantage because you are sure where you put your label. But the fact that is unique may be also a problem because you have no freedom. So then you cannot label the protein elsewhere. This group is only there, and so yes, that's something to be careful. And so these are some common strategy of labeling. Some warnings. Of course, you have to check the stoichiometry. So that means that you have to be sure that you have a level per protein, and that you maybe want to purify your sample again in order to separate the protein that were successfully labeled from the one that are not labeled. And so just to say that controls are necessary for being sure how your sample looks like, and to how to then proceed with the further investigation. Okay, so then I guess that I will stop here now, and what we say that we meet at 12:15 would be fine.
How much?
12:15, 15 minutes is too much, or I don't know. I mean, I will try to. I think that today I will stay. I will finish at 1 o'clock. I will not. Even with the break, I will not get too much delay.
Is it perfect? So
Is perfect?
yeah.
Okay.
So we'll meet at 3:45 here. 3:45.
No, at 3 at okay.
15 minutes. So, okay.
Okay, for you, yeah, sorry,
Yes,
I was
yes.
saying, yeah, sorry. Yes, so let's say, yeah, 15 minutes, yeah,
Yes.
at your
So
45 and mine 50.
yeah. So
Okay,
15
so
minute
then,
break, and we'll start at 3:45 p.m. Okay. Alicia will again.
okay, perfect.
The,
So see you. So actually, what is better to do, that I disconnect and reconnect, or what is it's maybe better, or what is the procedure? Last time I think that I remained connected, but I don't know what's the best.
It's up to you. If you the participants, they are online, so it's a 15 minutes time. It's up to you if you want to reconnect.
Okay. If not, what I can do is just I switch off the video and the microphone. So just
Yeah.
I put me
Perfect.
silent, and so then everyone is free to do, voila.
Perfect.
Perfect.
Okay. So, hello again, everyone. Hello?
Yes, yes,
It's
Alicia.
fine?
Yeah.
Okay. Okay, so then let's go on. So where we were? We were here. Yeah, okay. So we have just seen how we can introduce organic dyes as so fluorophore on protein by traditional bioconjugation methods, and also importance, the warning, if you want to, especially if you want to have quantitative information. So it's required to know how many proteins they are really labeled, and actually ideal to have only the protein, the labeled protein. Okay, I go in. So there are also another way to tag a protein, and so actually let's focus for the moment only on the left part of the slides. So where is practically a sort of overview of protein labeled, let's say, but with protein label in the sense is not only that we label protein, but that we label the protein with using actually a protein or a peptide. So so what we have talked about already, and so here is an example. So we can actually label our protein of interest, which is in pink here, with a fluorescence protein, GFP, for example, of one of the variant according to the property that we want to study and the wavelength that we want to focus on, and so on. And so, and this is a molecular biology issue. So we just we create a plasmid where the gene of the protein of interest is there together with the gene of the fluorescence protein. And then both protein together are expressed. So practically it's like we expressed a new protein that has two domains. One is the protein of interest and the other is the fluorescence protein, to visualize it. So there can be also done with a peptide, so similar let's say similar method. So just we instead of a fluorescence protein, we introduce a peptide that can then interact with a fluorophore. Okay, so then it's again we need a modification of the plasmid, and then the peptide. Maybe the peptide has already the actually the fluorophore integrated, and so yes, that is the difference between the two. It's, for example, there is a difference in dimension, and for certain purpose can be more interesting, and then it depends of your system and what you want to what you need, what is better. So you have just to consider several options, and So, and then there is the protein tag in the sense that actually that is a new, let's say, a new method, a relatively new method, which quite has some is practical. So there exists some self-labeling protein. So, and in particular, so for example, here is the mechanism. So that belongs to this category. Snap and clip. So then snap and clip are two self-labeling protein in this sense. So let's focus, for example, on the case of snap. Then it's quite similar. So the snap is an enzyme that has a substrate as a guanine. So what is done? How is done? So first of all, you need to express your protein of interest. Here, in this case, is green. With together with the SNAP protein, and then is added the fluorescence probe conjugated to a guanine, and this enzyme can convert this substrate, and the product at the end is that the moiety carrying the fluorophore stay covalently attached to SNAP and so to your protein of interest. The mechanism of clip is quite similarly, simply that the difference is that clip is reacting covalently with the dyes conjugating to cytosine instead than to guanine. Once the protein tag has covalently bound to the ligand, no other ligand can be bound as the catalytic center is blocked. So that is a quite efficient way of labeling a protein through a protein, practically. Yeah? It's not with the protein, but through a protein. Okay, so yeah, another method, but that we were talking about just in the previous slides, is how to incorporate a unnatural amino acid. That is so here there is no the intermediation of an additional protein, so is maybe more convenient in certain respect because you don't add a large new element in your system, and of course again it depends on what you have to do and what are your freedom area that you have, how to play with your system. Okay, so concerning label with the fluorescent protein, definitely GFP was opening this chapter some decades ago. So and the GFP was welcoming really with great enthusiasm because practically was the labeling was relatively simple. And especially due to the fact that it's based only on biomolecular biology, molecular biology methods, you can also label inside the cell already, and it can be also used inside the cell. Cell, yes, but also it was also used in vitro. And so, for example, here is an example. Is calmodulin, this protein that we just cited before, with the 4 calcium domain, and which was labeled with CFP and the yellow FP. So then cyan and yellow FP. So the two variant, one of the first 2 variant of GFP some years ago. So how is working here the story? Since calcium binding imply a large conformational change of the protein, which imply the closeness of the extreme. And so you see the protein when without calcium is quite stretched, and then it's get much more compact when calcium is binding. So this process was inducing the closeness of the CFP to the YFP. And these 2 proteins, the color, the spectral property of these 2 are done that CFP can be donor for yellow FP in a FRET mechanism. So practically, when without calcium, the protein is stretched, the fluorescence protein are far apart, there is no transfer. And with calcium, the calmodulin get compact and imply the closeness of the 2 fluorescence protein. And so FRET is allowed. And that was the way in which calcium binding could be detected. The study was calibrated in vitro, of course, this system, and then actually is a system that can be also used to detect the calcium concentration inside the cell, as it is. So, yeah, it's good, nice, and of course, one critic that one could say yes, but actually the level, in this case, the level is larger than the protein that we want to study. So then I can understand some skepticism about this method, which works and it gives good result, but maybe it's just for detecting the presence of calcium more than to have really detailed mechanistic information about the protein, the calcium binding protein itself. So fluorescent property, in fact, so practically we touch the one drawback of fluorescent protein is the fact that they are very large levels. And moreover, they have, some of them have a tendency to oligomerize, and so introducing additional complication to our system. But they are well adapted to cell imaging. And so, again, balance between advantage and drawbacks and choose what is the best for you. Another, since we were talking about and also shortly about nanoparticle, in particular quantum dots, we can label protein also with nanoparticle. And yeah, the quantum dots are powerful labels, as we saw. The brightness is quite large. But of course, they are even larger than fluorescence protein. So again, it depends on your system. Moreover, you may enter into troubles when you want to purify your sample to separate labeled and unlabeled proteins, because size exclusion methods do not apply because of the large size of the functionalized quantum dots. So then you have to find adapted way of purification. So additional efforts are required, but it's definitely an option. Yes, actually, I ask you, maybe I will use some minutes still, to start the next lecture. So again, because in order to be sure that all what I want to say, since tomorrow is the last hour, the last session, and so I cannot catch up another time, and so then I have to try to fit in everything. And so if it's okay for you, I can just start to introduce the next topic, that would be the fluorescence probe in amino acid. And yeah, or if not, I stop here. So that is up to you.
So so I think you have finished for the fluorescence probe for protein. So I think
in principle, yes, yeah.
Then it's okay. I think we can start with next day. Yes, next day with the fluorescence protein for the nucleic acids.
Okay,
Yeah,
so
yeah.
then it is so I will try to keep time in the to stay in time, of course, tomorrow. Tomorrow I think that I am planned for 1.5. Is it true? Is it the classical morning? So for me, the classical morning session, and so okay, good. So then today we exceptionally end a bit earlier.
Yes. So student have any questions, queries?
Comments also.
So any
can,
comments? Yes.
Yeah, that's it's don't hesitate. It's we can discuss also about something.
So
No.
Simran, are you here?
Yes, sir.
Yes.
Thank you so much, ma'am, for such a nice talk.
Okay, good.
I wish you all found this lecture very useful and informative. So I want to thank everyone for your active participation and enthusiasm throughout the session. And a special thanks to our esteemed instructor, Dr. Jagannath and Dr. Aliza, and for their talk. Sir, should I close this event now?
So yeah, so then practically I will say bye. So we see tomorrow. So that's the point. Is okay? Yeah.
Yes.
Okay, good.
So I formally bring the today's lecture to a close, and thank you once again, everyone. And we will meet tomorrow again at 2 p.m. At 10 a.m. And our next speaker would be Dr. Pamita Vasti. So we will meet again at 10 a.m. tomorrow.